because the fatty acid chain is very long, it's also very low in terms of oxidation state. So when you burn it, it has more levels of being oxidized. So it's further down, and then you can oxidize it all the way up, so you can get a lot more energy. That's a big difference in that. So that's why we prefer to store fat, or the cells prefer to store fat, not us. We don't prefer that. But the cells definitely prefer to store that for you, because that's a more secure form of getting a constant supply of energy, basically. Now, and reliable supply. The other thing is that if you don't have the storage, then you can hopefully get it from the diet. And if you don't have it from the diet nor from the storage, then the cells will make it for you. And this is how I'll show you how the cells will make it. So the synthesis of fat is referred to as lipogenesis. So lipogenesis, we know that fat is actually made up of glycerol esterified to 3 fatty acids. That's because that's what we call triacylglycerol. But the starting material for making fat is not glycerol, but glycerol 3-phosphate or DHAP. Okay, that's just the nature of the thing. So although this is fat, the starting material for making fat is glycerol 3-phosphate or DHAP. And of course, DHAP is the same thing as when glycerol is converted to glycerol 3-phosphate, and then you get DHAP. You have to know what DHAP is by now. DHAP is when you split glucose. You split it into DHAP and glyceraldehyde. So you see how the previous chapter is so important in building this chapter. So that's the starting material. And then what you do is you put in the fatty acid, one at a time. And you make your triacylglycerol. And this process of making triacylglycerol is of course referred to as lipogenesis. Alright? So let's do the whole pathway. So I give you the overview very quickly, what is necessary. And of course when you go through the pathway, it's painfully detailed and difficult. Let me turn off the lights to get better contrast. Did it improve at all? Do you think it improved? Just makes you go to sleep, right? Turn off the wrong side. Is that better?
Yeah.
Huh? Okay. So basically, I told you, your starting material can be glycerol 3-phosphate or DHAP. Now, because DHAP can become glycerol 3-phosphate. Do you see that? That comes down. Glucose to DHAP is part of your beginning part of your glycolysis. And then you split glucose into DHAP and glyceraldehyde. And then the DHAP can be converted to glycerol 3-phosphate. Or if you got the glycerol from the breakdown from your storage of that, glycerol can come to the liver and be converted to glycerol 3-phosphate. So you got all kinds of sources of glycerol 3-phosphate. Glucose can become DHAP, which can become glycerol 3-phosphate. The breakdown of fat, when you take the fatty acid to be burned, further for energy, the glycerol part can come back and convert to glycerol 3-phosphate. So we got all the materials that we want from all kinds of either recycling back or even from glucose. So even from glucose, we can make fat out of it. That's what I'm trying to tell you. So that's your starting material now. It's going to go into the mitochondria. So now you have to keep track of, so this chapter or this section is a little bit more challenging now because you have more organelles to worry about. Before that, it's only mitochondria, cytosol, mitochondria, cytosol, and so now you see that this thing can happen in other organelles as well. So, but the main part is in the mitochondria, the most commonly done way. So we go into the mitochondria, and then in the mitochondria, we're going to add the fatty acid to it consecutively, one after another. So not all simultaneously, but one after another. So you see the fatty acid is attached to CoA as a carrier, and then it attaches. Once it attaches to this glycerol 3-phosphate, then you get coenzyme A being remade back. And then you got one fatty acid chain. So you do that again, and you get the second fatty acid chain. Do you see that? So you're adding consecutive fatty acid in the ester bond. And then the last one, now, if you recognize this is a phosphate with a glycerol with 2 fatty acid chain. What do you think that resembles? We've done this before. What do you think it resembles? It's a phosphate with 2 fatty acid chain. It's your phosphoglyceride. That means it's one of those things that you can use to build your membrane bilayer. So at this point, we were supposed to be making fat, but at this point, if you have some membrane structures that need replacement, your phospholipids needs replacement, you can actually, take some of these to make your membrane or to fix your membrane structure. If you don't, then we can continue on. It's not like you're going to take everything. A certain percentage goes towards repairing your membrane. The rest of it goes further, and now you remove the phosphate. So now you got 2 fatty acid linked to a glycerol. It's a diacyl glycerol, and once you remove that, you can further add the 3rd fatty acid chain to it. So basically, you start with this, and then you consecutively add fatty acid chain to make your triacyl glycerol. And this is done in the mitochondria. This is the main activity. Now, other organelles can also make fat, and here you would see that they share some of the features. I just want to get out of that black. But I want to see, you see that? There's 2 other organelles that also does the fat, and the starting material is different. In this case, I consider glycerol 3-phosphate as the starting material since it goes into the mitochondria, and that's when you start packaging it to fat. In the endoplasmic reticulum and the peroxisome, you start with DHAP. So that means you instead of the DHAP becomes glycerol 3-phosphate, you just take DHAP and start adding fatty acid chain here. Do you see that? That's a slight difference. So you add the fatty acid chain to DHAP, and you have this intermediate here. And then after that, you reduce that, and it comes back to look the same here. So in this case, we add the fatty acid to glycerol 3-phosphate. Here we add the first fatty acid to DHAP, and then convert that to the same method. And then everything down from here is the same in terms of the pathway. So these are the 2 different, so different organelles might start differently. That's what I'm trying to say. Different organelles will start adding the fatty acid to glycerol or to DHAP. That's how you're starting. So that's how I summarize it for you. Now, this process of making fat takes place in the liver. So now you also have to know the organs. that makes the thing. When you're studying, you have to be aware not only the name of the pathway, what is the starting material, what are you trying to make, which organelle and which organ you're taking, these things are taking place. So this is taking place in the liver. You can do it also in the adipose tissue. So nice lipogenesis for you. So let's look at lipolysis then. Lipolysis is the reverse, right? Lipolysis is the breakdown. Now breakdown is slightly different from making. So this is lipogenesis. That's what I did just now. So now I'm going to do the breakdown. When you break down triacylglycerol, it's much easier. So you have the glycerol esterified to fatty acid, and it's very easily just breaking all three ester bonds. Yeah?
Can you use a different color? I can't see.
I can't see the red.
So it's, remember, triacylglycerol fat is linked through ester linked to three fatty acids. It's just breaking the three ester bond, so you end up with glycerol and three fatty acids. So it's much easier to break down than when you build it, basically. And that process is called lipolysis. Alright? And of course, breakdown can occur where? In the adipose tissue when you are storing that. Okay? Now, to break down, you need signal. You need the signal, glucagon and epinephrine, to tell you that, you know, just like when you're breaking down glycogen for glucose or glucose 1-phosphate, you need signal. And the signal is in the form of glucagon or epinephrine binding to receptors. They don't go into the cell, they bind to receptors. And that binding will then activate a hormone-sensitive lipase. Remember, we went through 3 different lipases, and I told you, you have to know the 3 different lipases and what they do. They do it differently. For exam, you've got to know that because you never know which lipase I'm talking about. And they behave differently, so you have to be able to answer the question. The hormone-sensitive lipase is the one that is going to hydrolyze. In this case, when glucagon and epinephrine activates, they activate a hormone-sensitive lipase. This hormone-sensitive lipase is also referred to as triacylglycerol lipase. So it comes in 2 names. You have to know both of them. All right, so these breaks down into glycerol and fatty acid. The glycerol again shipped to the liver because we can reuse the liver. Reuse in the liver, we can reuse it to make glycerol 3-phosphate, which then goes to making fat. All right. The fatty acid. This is the useful part. This one gets bound in the blood to albumin, serum albumin, and then it's taken to all the muscles, particularly the cardiac muscle. Cardiac muscles don't have downtime. You know, if you do, that is the end of you. They have to constantly be beating, right? Your heart has to pump all the time. So it has to rely on a reliable source, a source that is not going to be depleted easily. It's going to be always able to supply energy. So that's why it needs the fatty acids. All right? And now, once it reaches the cell, what do? In the blood, is carried in, bound to albumin, and then when it reaches the cells, it gets transported into the cell, and then it gets bound to fatty acid binding proteins before it gets to be incorporated into the different organelles. So it can go into mitochondria, endoplasmic reticulum, and it can also be used in the peroxisome. So it's all connected. Do you see that just now? When we were making fat, the fat can be attached and then goes into the mitochondria, or it can go into the endoplasmic reticulum, or it can go into the peroxisome, where you can again attach to glycerol 3-phosphate or DHAP. So they're all connected. That's only if you're making fat. But if you want to further break down the fatty acid, then you do it differently again, right? So these are lipolysis. So you know lipogenesis and lipolysis, right? This is just a cartoon look at what happens inside. I went through, I told you that for lipolysis, you need the signal. The signal is glucagon and epinephrine binding to the receptors. This is the adipose site, which is the cells of the adipose tissue. And here is your storage of fat, basically. The storage of fat is in there. So the signal binds, and then again, you have the same kind of concept that it's not any different from when you were learning about glycogen breakdown. This is a little bit shorter. We don't have that big cascade that we deal with in glycogen. Here, it still wakes up the adenylate cyclase, the enzyme that makes your second messenger, your cyclic AMP. The cyclic AMP wakes up protein kinase, and when protein kinase wakes up, what it does, a kinase adds a phosphate. So it adds a phosphate to wake up the hormone-sensitive lipase. That's what I was mentioning here, your triacylglycerol lipase. This is the same name the same enzyme but named with two different names, all right? This is your hormone-sensitive lipase or triacylglycerol lipase. So when you add a phosphate to it, it's activated, and once that's covalent modification, it's activated and it starts to break down or hydrolyze your fat into, it removes one fatty acid chain, then it removes a second fatty acid chain. Do you see that? And then it removes a third fatty acid chain. And you end up with glycerol and 3 fatty acid. This fatty acid goes into the blood bound to serum albumin, and then it can be, then in the cell is bound to fatty acid binding protein, and then it can go into different organelles. All right? So you understand that? Now we want to see what happens to the fatty acid. So I taught you to take fat and how to make it from glycerol or DHAP depending on which organelle you're using. And then I taught you to take the fat that is stored in your adipose tissue and how to break it down to glycerol and 3 fatty acids using your hormone-sensitive lipase and how the glucagon and epinephrine is used to activate that. Now I'm going to go on and show you how to deal with the fatty acids. Remember, this is the valuable part. I mean, they're all valuable, but this is like the cream part that you want, because this is where you want to break it down to lots of energy. All right? Now, the process of breaking it down can happen in the peroxisome, or it can happen in the mitochondria. Mainly in the mitochondria, the preoxysomes deals with the longer one. So remember I told you this fatty acid can be C18, C16, C18, all kinds of mixture. Sometimes you have the extra long one. The extra long one are like the C22, C20. Those are extra long. The extra long ones usually gets to the preoxysome first, because the mitochondria doesn't deal with the extra long. They tend to deal with the long and medium chain. The extra long ones goes first to the preoxysome where they trim it to about C16, C18, and then they ship to the mitochondria. So they pre-trim it if it's very long. But the common ones are usually C18, C16, C14. Those are dealt with directly with the mitochondria. That's not an issue. All right? Now, to get into the mitochondria, they just can't just walk right in or diffuse right in. There are the inner mitochondrial membrane that is very selective. I've already told you that. So in order to get in, remember fatty acid has one end that's charged. So it's not going to go through a membrane easily, so it has to be transported across the inner mitochondrial membrane. It has to be transported in so the transport system is actually a protein called carnitine. To allow it to go in, it has to be handed to this protein that will then take it across. So there's different types of transport, right? Now, the fatty acid must first be activated, and what that means is it first to be attached to CoA. CoA has a high energy bond, so it's activated by binding to CoA first. And then CoA would approach this transport system, this protein, and then hand over the fatty acyl chain. So this is a fatty acyl. When you call that, this is fatty acid. Do you see that? But like that, it's called fatty acyl. That's your organic terminology again. So this is a fatty acyl chain. It's linked to CoA, but then when CoA brings this fatty acyl CoA to carnitine, then the acyl part goes into the carnitine to make acyl carnitine, and CoA comes back out. Then it's transported across because it got a carrier. It's like a thing on the boat. It's allowed to ship it across. When it reaches the other side, it gives it back to CoA. So it's only used as a transport system. So CoA is on the other side, is waiting, and it hands over, and then you end up with fatty acyl CoA. So it looks the same from inside and outside, but it has gone through a transport system. Do you see that? And now this is attached to a CoA process. So we are inside. That's how we get inside. And then the process begins. So you see, this has several carbon, and this is the hydrocarbon portion that is going to be broken, or that's going to be, what we say, oxidized. Because this is very low down in the energy, you can oxidize it many levels to create a lot of energy. But we cannot do it all in one step. So it has to be done 2 carbon at a time. And then it's cleaved, and then you cycle. So you do as many cycles as you need to complete this. So the longer chain, the longer the cycle, because you can only manage 2 carbon at a time. Do you see that? So let's do the process. Once in the mitochondria, this process of metabolizing the fatty acid is called beta oxidation. Oxidation, do you see that? First step is an oxidation process. And that oxidation process means I oxidize this. Everything about getting energy is oxidation. Even glucose in glycolysis is oxidation. So this is one oxidation step. And the first step creates FADH2. Is it good or no good? Is FADH2 of any use to the cell or is just garbage?
It's an electron carrier.
How much is it worth to the cell? Huh? Yes, 1.5 ATP, especially if it's in the mitochondria already. Do you see that? So it makes sense that we are in the mitochondria because they can be translated into energy. Where? Where in the mitochondria is it translated into ATP? Electron transport chain. See how that previous chapter is so important still? Otherwise it's meaningless, right? So you have a fatty FADH2 that comes out of the first step. We only step 1 and you're already making 1.5 ATP. Step 2 is just to add water. It's like cooking. Step 3 is another oxidation. And this time, step 3 creates one FADH. Is it better or worse? Huh?
Yeah.
Better. How much better? How much ATP? Not 1.5 anymore. Huh? No, not 3. You round it up. How much is one NADH worth in the mitochondria? 2.5. Yeah. See, you're counting, right? And it's only step 3. You're already gaining. You're thinking, wow, you know, it's worthwhile. And then the last step is a hydrolysis. So you break. Oops, I break the wrong. You break it. And you break it. And you get what comes out of that break? Acetyl-CoA. Do you see? Because I break it at the 2 carbon at a time. Is acetyl-CoA worth anything in the mitochondria? No? Wow. Why are we in there? What did we do with acetyl-CoA in the mitochondria?
Citric acid, sir.
Huh?
Citric acid, sir.
In the TCA cycle, what did we do in the mitochondria with acetyl-CoA?
Bonding with acyl acid.
We break it down, right? In the mitochondria, how many NADH did you get from one cycle or from one acetyl-CoA in the TCA cycle? No? 3 NADH. How many FADH? 1. How many GTP? 1. So how can you say it's no use? So we are in the mitochondria, so when it breaks down, it just goes to the TCA cycle and you can give out 3 NADH and then 1 FADH2 and 1 GTP that you can use as energy directly. That's already just step 4. You can gain so much. Of course, you have to add step, the amount from here and here too. Do you see? And depending on how long this chain is, we're going to go another round. And the second round, we're going to start again. So after I cut this part attached to a fresh coenzyme A, this comes out as CoA. This is shortened by 2, and it goes through another cycle. And each cycle, the first step is you got 1 FADH, 1 NADH2, and then eventually 1 acetyl-CoA. So if you do many cycles, because this is a very long hydrocarbon chain, then you're going to make many FADH2, NADH2, and acetyl-CoA. Now do you agree that this is a better storage even though it doesn't make us look pretty? That's why the cells wants to store. The cells don't care if you look pretty or not. They just want you to live. You use it to survive. So that's why we store this in preference to, we do store glycogen, but we know that you cannot rely on glycogen for a long time. Do you understand? All right. So you're doing the cycle one at a time. And this is another picture to show you. You see that? So you come in with of course you started off with the acyl-CoA on the outside, acyl-CoA, the acyl group gets transferred to carnitine, which is your protein that translocated inside, that's your transport protein. And then once inside, it gives it back to CoA, so you end up with the same looking inside and outside. And then you start the process. The first step of oxidation create FADH2. This is created, this is produced, but you need FAD. So what you need is FAD, what you make in the first step is FADH2. Second step, you just add water. Third step, you need NAD+, but you make NADH. Third step is when you lyse it with the help of coenzyme A. You add the CoA, so you lyse this. This CoA comes out, and then a new CoA attaches to the shortened one. So one coenzyme A comes out, and then this part gets attached to a fresh CoA. Do you see? And the cycle goes on again. So you do as many cycles as it takes to break it all down. So you break it down to FAD, NAD, and one CoA, each cycle. Now, so how many CoA would you get if you have a C10? Remember?
5.
It's 2, 2, 2 at a time. So how many? 5 is correct. But how many cycles do you do to get to the end if you have C10?
2.
Huh?
Do 5 cycles.
No.
2.
No, 2 means you only cut here. Do you see? You only cut twice.
10.
Keep going.
10.
Look. So you cut once here.
Oh, 4.
You cut one. Yeah. Why do you only cut 4 if you get 5? Because the last cut is like cutting a slice of cake into half. You get 2 pieces. Do you see? So you actually cut 1 less than what you get. So if you have a C10, you have 5 acetyl-CoA because it cuts 2, 2, 2 at a time. But then the last cut, you gain 2 pieces. So you only need 4 cuts or 4 cycles. So for a C10, you only need 4 cycles or 4 cuts. Do you see that? So then if you have 4 cycles, then you multiply all these by 4. Oops, sorry. Yeah, you multiply those by those by 4. This one, we know that this is going to be 5. You divide by 2. Do you all get that? Okay. But you can see there's so much from even the C10. I didn't even go to C18. So C18 you get more too. But you see, because even each acetyl-CoA, you're going to get 3 NADH, 1 GTP, and 1 FAD. So that 5 is going to be multiplied by 3 NADH to add to this, which is multiplied by 4. So you can see why storing, and that is only 1 fatty acid. How many fatty acid is in a triacyl glycerol? 3. Do you see? And so you can get so much more out of it. the fatty acid breakdown of saturated. See, these are all no double bond. However, if you have a double bond in the process, then what do? Well, you do everything the same before the double bond. So that means ahead of here, everything is treated the same way as you would treat the single bond. But once it comes close to where the double bond is, there's one enzyme step, and that's all you need to know. There's just one enzyme step that converts that double bond to look like before. So all you need to memorize is this name here, the NO-CoA isomerase, to switch it to look like the saturated form. So it only happens when you're approaching close to this. So after you cut these, and when you're nearby, there's one enzyme to switch it. One enzyme steps just to correct for that. That's all you need to know. So that is beta oxidation. Everybody knows beta oxidation. And where do you think it happens? Yes, mitochondria. That's your beta oxidation. So now that we do beta oxidation, I have to teach you how to make fatty acid. Because we always either break or make, right? So now you know how to break down fatty acid, but you know how to make fatty acid chain out of what is the starting material you think would be a good starting material to make fatty acid? What do we break it down to? When we break it, we break it to what? Acetyl-CoA. So when we make, should we also be able to make it out of acetyl-CoA? Alright? So basically now it's the same. When you break it, we went through cycle. When we're attaching it back up, when we're growing the chain back up or making the chain, extending the chain, and we start with acetyl-CoA, we should also do it by cycle. Except that most things are going to be the reverse. When we break it, it's oxidation. When we make it, it's reduction. All right? So the process now. Just to tell you that, it's always like that. It's never that straightforward. You can't just glue things together as straightforward as that. Not in chemistry. Maybe in some reaction, yes, but then I also want to show you where you can build the fatty acid from. We already established that you can use acetyl-CoA. Since you break down fatty acids, you get acetyl-CoA, then acetyl-CoA can be the starting material. But remember, where did you get your acetyl-CoA from?
Glucose.
Huh?
Glucose.
Yes, from glucose. When you took glucose and you go through glycolysis to pyruvate, and then you enter the mitochondria, and you make acetyl-CoA through that multi-enzyme complex, and then you're in the mitochondria. But I'm telling you, the synthesis, in the mitochondria we have beta oxidation. So we don't try to crash the two. We try to separate the two processes. The breakdown and the building should be in two separate organelles so that they don't conflict with each other. So I'm saying, yeah, it's nice that acetyl-CoA is in your mitochondria, but the process of fatty acid biosynthesis is in the cytosol. So, can we just drag acetyl-CoA out of the mitochondria? Acetyl-CoA can't come out of the mitochondria. So what is the option? Citrate. You make citrate. And it's not that difficult. Why? Because it's already, you're in a mitochondria. If you make citrate, that's the first step of your TCA cycle. And every time you build up a lot of citrate, it means you have a lot of energy, because that's PCA cycle is about making more energy, breakdown of acetal. So if you have a lot of energy, then you have the means to build fatty acid, to make something out of it. So, but citrate can come out of the mitochondria. That's why, acetyl-CoA cannot, oxaloacetate also cannot. Remember, we had converted to malate and then shuttle it out. But if we go forward and we make citrate, citrate comes out through the tricarboxylate transport system. And once citrate is out, that was the pathway the transport that I tested you all on. When citrate comes out, it reverses to acetyl-CoA and oxaloacetate. Remember that transport system? And then of course this stays outside. Why? Because we're going to make fatty acid out of it. This one, we can take it back inside by becoming malate. And malate can go in. Now, if we don't want malate to go in, we can make this into pyruvate. We can make this into pyruvate. And this is an advantage, because anytime you make this into pyruvate, you also make NADPH. Where is NADPH also made earlier? Do you remember? Anybody remember? We did talk about NADPH. There is one pathway that makes NADPH. What? Pentophosphate pathway. The ones that don't make ATP, make ribose 5-phosphate, also make NADPH. Let's say if you already spent it, then you don't have enough, or you need more. That's when you take oxaloacetate to malate and malate to pyruvate and you make more. Why? Because I need it for fatty acid synthesis. So it's appropriate to at least check the storage and say, oh, I need more. I'm not going to let malate go in through the shuttle. I take malate to pyruvate and I make NADPH on time to make fatty acid. All right? So now we're going to make fatty acid. Do you see that? So I'm going to take, this is just the chemistry way of putting things. It always oversimplified. You take acetyl-CoA, you need the NADPH as you're reducing. Remember, when we did beta oxidation, it was oxidation. But now it's the reverse. Building is the reverse of breaking. So now it's a reduction process. And to reduce, I need NADPH. The enzymes in fatty acid synthesis need NADPH. So I need NADPH, and I'm going through several cycles, and then I make the chain, the C16 chain. All right? Right? Because if I have 8 acetyl-CoA, and each acetyl-CoA has 2 carbon, then if I string them all back together, I should be able to get altogether 16 carbon, and that is your palmitic acid. All right, and I need 14 NADPH to do it, but I only need to go through how many cycles? It's the same logic here. When we broke it down, we always use 1 cycle less than what we need. So 7 cycles is all we need. Although we use 8 acetyl-CoA, because when you attach 2 together, that's already 1 cycle, and you start with 2. All right. Now, wish that was so easy, like the chemistry way, where they just give you an equation. Well, this is how it's done in the cell. I have to give you the whole pathway of how it's done. So, now when you want to attach something, you can either take two carbon and attach it. But in this case, it needs a 2 carbon to attach to a 3 carbon. And then during the attachment, one of the carbon leaves, and then you get 4. So this is how it starts off with. The top part, you start with acetyl-CoA. To build your fatty acid. I'm going to convert part of my acetyl-CoA, this is a 2 carbon structure, to a 3 carbon structure called malonyl-CoA. And then I'm going to require the multi-enzyme complex. This is a multi-enzyme. And the enzyme is a complex that can make 2 strands simultaneously. On this side is an ACP group with the SH. And here too is an ACP with an SH. So we were using the carrier as carrier CoA. I'm using as carrier initially CoA. That's on the outside. But when I'm going to do the synthesis, the biosynthesis of fatty acid, I have to change carrier system. That's just the nature of it. The CoA gets attached here. The CoA. Acetyl-CoA, it transfers the 2 carbon to a new carrier. This is my acyl carrier protein. So for biosynthesis of fatty acid, the carrier is not CoA, but acyl carrier protein, short ACP, and sometimes ACP, SH. It still has the high energy SH one, okay? This goes here. See, they're side by side. I'm just representing it. One go here, one go there. This is a 3 carbon chain. All right? Now we're on the multi-enzyme complex. And that's the process starts. So this is not yet. This is just getting ready to transfer it to the multi-enzyme complex. This is your enzyme now. And the enzyme does it simultaneously with this end as well as this end. So it can make fatty acid chain much faster since you can do 2 copies simultaneously. Now, the first process that takes place requires the enzymes will take this and then attaches here. All right? And this thing comes off as CO2. You see that? Now I have 4 carbon, and this is gone from here. And now I'll do the reverse step. Remember with beta oxidation, I did an oxidation, hydration, oxidation. Everything is reversed. I do a reduction. And who's giving the NAD? Who's giving me the hydrogen? NADPH. All right. Then I do, that one was hydration, so it's dehydration. The next step, just losing water. And then this step 1, step 2, and step 3, another reduction. And what is providing the hydrogen? NADPH. Do you see that? So we use 2 NADPH per cycle. All right, that's 1 cycle. And after the cycle finishes, then this is arranged nicely. This is then transferred back to here. There's a transferase that attaches here. Transfer it back to that. But this is processed. Do you understand? So before it wasn't processed, now after this reduction, dehydration reduction, it's a processed form. So it's the hydrocarbon. And then here, what comes in here? Malonyl-CoA, new one. Do you see that? And then, what does this do? Cut. So the cycle repeats itself. So then write this chain down to here. This becomes CO2. This one now becomes this. Do you see that? Oh, and then this is gone. And then I do the step again. So each cycle I'll do a reduction, dehydration, a reduction using these. That's cycle 2. And then I transfer this back here. And then what comes back in here is a new malonyl-CoA to be added. So you add using the malonyl-CoA. Do you see? But you only add 2 carbon at a time because it transfers and it breaks here, CO2 comes out. So you go through 7 cycles. If you have 8 acetyl-CoA, you only need to go through 7 cycles. You all get that? And then if it's 7 cycles, because each cycle you have 2 NADPH, then you have actually 14 NADPH. The 7 cycles, 2 each cycle. Do you all get the process? And then of course once it's C16, once it reached the maximum that it wanted, then it will just cut it off and that flows out. When it reaches the correct length, and you get your fatty acid biosynthesis. Do you all follow that? So what is the similarities and differences now between the 2 systems? When you have beta oxidation versus fatty acid biosynthesis. What is the similarities and what is the difference? Yeah?
Well, one difference is that beta oxidation occurs in mitochondria.
Yeah, one is in the mitochondria and the other
Such.
in the cytosol. All right. And then what else? What is the carrier system here?
NADH.
The carrier. The fatty acyl is carried on what? When it's activated, what is not the transport, but what is on the fatty acyl? Huh? Yes, acetyl-CoA. CoA is your carrier. So that's CoA here. What is your carrier here for the breakdown? Huh? What was the carrier? One is CoA, what was the other one? What was on the enzyme? When you transfer it, what did you transfer the fatty acid chain to be? Here the fatty acid chain is connected to CoA during the whole process, each time. Even after you cut it, you still connected to the shortened part is still connected to CoA. I ran out in here too. This is connected to CoA. What was this connected to during the whole biosynthesis? What was it attached to? ACP, acyl carrier protein. All right. So this one also requires a multi-enzyme complex. This uses single enzyme. That's another difference. This one is to break down fatty acid into 2 carbon. This one you build up fatty acid using a 2 carbon and 3 carbon. Actually using a 3 carbon to add on the fatty acid. So that's just the differences and similarities that you need to be aware of. How much time do we have? Now, I told you about making fatty acid right from this biosynthesis. You make it with acetyl CoA and then as malonyl CoA, which you then transfer to acetyl ACP and then malonyl ACP. And so this is your new carrier. And you keep adding 2 carbon to your acetyl group using a 3 carbon basically source. Now that is the long way of making fatty acid. You can, right from the very beginning, from scratch. You can also do the shortened way, where you can start with a shorter branch or shorter chain and then add on. So there is no rule to say, oh, you always have to start that small. Most of the time you would use it that small, but you can also take like a C10 and elongate it. That's what I'm saying. You can do shortcut to it. So if you want a C18, you can start by elongating a C10. And that occurs in the endoplasmic reticulum, or it can occur in the mitochondria. When we talk about lipid, the endoplasmic reticulum is not the rough endoplasmic reticulum, but it's the smooth endoplasmic reticulum. So basically, I can start with a C10 and then elongate that to a C16 or C18. Now, I can also make double bond. Remember, if you have the right enzyme, the enzymes are called desaturase. Animals can make double bonds at carbon 9, means 9 and 10. 6 means 6 and 7. 5 means 5 and 6, basically. That's how, they only showed you the lower number. So these are the 3 positions where we have desaturases for it. So if you want to make double bond, I'm talking about animal system, not plant, then we can make double bond from the saturated form that we have. But the double bonds are only at fixed locations. We can't do past 9, remember? We can't make linoleic acid or linolenic acid. Plants can, but we cannot, because we're only limited by the type of desaturase we do. Now this process of making double bond is very similar to the electron transport system, but it's not. It's happened on a membrane structure too, and it's also drawn by oxygen. So basically, remember the electron transport system is operated by having the electrons coming from NADH and it's drawn to oxygen. But it's not the same. It's just very similar kind of operating idea that you can get enough energy from making a double bond using a kind of an electron donor acceptor kind of concept.
So this only happens on saturated?
Pardon?
Desaturation only happens on saturated ones?
Oh yeah, desaturation is when you have saturated fatty acid chain and you want to make double bond, but you can only make it at this location because the enzymes are only that specific. Now this one is the regulation. This is like the entire picture, the whole shebang. See, it starts all the way with glucose. So it's a mistake to think that, you know, glucose is separate when you're dealing with fat metabolism, and you have nothing to do with carbohydrate. Actually, it's all connected. So you can see that I can start with intake of glucose, and you can then take that glucose into the cell, and you can metabolize or oxidize it to produce pyruvate. And this part, of course, you remember them as your glycolysis. You can also take the glucose 6-phosphate that comes out of your glucose through the pentophosphate pathway to make your ribose 5-phosphate as well as your NADPH. Alright, you can't see here, it's NADPH that's made, which can then be used later on for lipid metabolism. So as far back as glucose metabolism, you are already planning or connected to lipid metabolism. You are somehow, when you have glucose, you are also thinking ahead. One day I'm going to have to store fat. If I have plenty, why not go through the pentophosphate pathway and make some NADPH ready? Now, I can use the NADPH for what? Regenerating glutathione. I can spend that first. I can save some for fatty acid. But I can always make more. Do you see? Remember, we made more even right before making fat through another way. So that's not a problem. We made some, we might use it, or we might save it. We take the pyruvate into the mitochondria. Once pyruvate goes into the mitochondria, part of it goes to replenishing your oxaloacetate. Make sure you have enough oxaloacetate. And then part of it goes to making acetyl-CoA. Acetyl-CoA will combine with your oxaloacetate through the TCA cycle, and you can get a lot more energy. And once you got a lot of energy and you build up a lot of citrate, then you say, why do I just keep making energy if I'm not going to build and store for bad times? So citrate comes out of the mitochondria. Do you see that? And citrate comes out of the mitochondria, is transported through the tricarboxylate transport system. Once it comes out of the mitochondria, it becomes oxaloacetate as well as acetyl-CoA, oxaloacetate, acetyl-CoA. This one can go back, not directly, but as malate, if you don't need to make any more NADPH, but if you do, you delay it by making pyruvate, you replenish some of the NADPH that you use up for maybe regenerating glutathione. Alright? And then pyruvate goes into the mitochondria again. So you can get that. The acetyl-CoA that you cap out here now is going to be used for your biosynthesis of fatty acid. Remember, it's outside in the cytosol. This one is in the cytosol. That's why you make some of it into malonyl-CoA, and then you assemble those two together to make your fatty acid chain. Now, if the scenario changes, all of a sudden, somehow you need to do a lot of work. And they says, oh, we can't store the fatty acid that you just made. Don't. I need to burn it for energy. And so, there you go. Take it back in there. You see that? The newly synthesized one, because you started, doing vigorous exercise. So now this is then made, transported to carnitine, and then carnitine then reverse it back to CoA, and you have beta oxidation to break it down to acetyl-CoA and get energy in. So the thing is all dependent on timing, what the cell needs, all the signal, whether you need energy or you have way much energy, then you start doing it. Now, in terms of activation, inhibition. This step that makes the fatty acid chain is inhibited by glucagon and epinephrine. So if epinephrine is present, it always says to break down. So then that process will be stopped. We don't have two opposing things happening at the same time. This is when epinephrine and glucagon comes in, it meant they said, oh, I need to harvest your fatty acid and break it down for energy. So this step should be inhibited, because if you're already breaking it down, why are you building at the same time? You can't do two opposing stuff. So basically you shut down this while you're breaking that down, because you want to reserve the energy now and use it for other things. This, on the other hand, is stimulated if citrate is plenty. So this is inhibited by glucagon and epinephrine because those signal break down. But building, if citrate is plenty, then it signals making that. The feedback to inhibit this is also to make excessive amount of fatty acid chain. There has to be a feedback. You don't want to make a lot. Cells don't like to make a lot and then let it deposit everywhere, because then it becomes a disease. They only make what is necessary enough, so you don't end up with excessive amount of something, and then you will end up with a disease condition. So whenever you make sufficient, then it will feed back to inhibit that step, because that's the step to making it. So basically, there's always a feedback and an activation step. If you need energy, then that is inhibited because this is to be taken into breakdown. On the other hand, if you have excess energy, then this is activated to make fatty acid, which can be stored or which later can be used for energy breaking down. So regulation is also an important thing because it tells you what can happen and what cannot happen. There are two types of regulation. There's a short-term one and there's the long-term. Long-term is very easy. Long-term is about, well, you have to go back to your genetics and says, okay, I need to have certain types of hormone being made because I want to trigger certain things. Those are the long-term effect. It goes back to your genetics and it involves making more of one type of hormone for breakdown or the more of the hormone for building, depending on the state of your cell. So that and your type of diet. So if you are always on a high-fat diet, then chances is that they're not going to activate the hormone to make more. Do you understand? Because you're already on a high fat. So it means then it's going to shut down those enzymes and hormones that are going to activate the synthesis of a lot. On the other hand, if you're on a low-fat diet, then it makes sense that, well, you're not getting sufficient from your diet, so I have to activate the enzymes in the pathway that are going to make more for you. So it just works that way. That's a long term, because it takes a while to activate a long-term one. The short-term ones are the immediately impact. That means what wakes up those enzymes that are already existing. They are already there. So glucagon and epinephrine, for example, they are activated by the short term. So, glucagon and epinephrine are the ones that activate the short term effect. The enzyme is already in the cell, waiting by the side of the storage. And of course, when glucagon and epinephrine bind to the receptors, then the cyclic AMP will be made. Cyclic AMP will be made, which is your second messenger, and that will trigger protein kinase to add a phosphate to your hormone-sensitive lipase, and then you will wake up and says, okay, I've been told to chop out the fat, into free fatty acid and glycerol. All right? So that's the signal that comes in. So that's the short-term impact, basically. And then, of course, the glycerol will be taken to the liver. The fatty acid will be taken to the cells other than the brain. Now, remember, brain don't use fatty acid. All the muscle cells will require the fatty acid to be broken down further. So that's to stimulate the breakdown process. Now, if they want to rebuild again, then insulin will be the one that comes out. And then, of course, the whole process would be about building the fatty acid. But, you know, glucagon and epinephrine is about breakdown. One more. Ketone bodies. What are ketone bodies? Ketone bodies are actually formed from acetyl-CoA in the mitochondria. You don't even need to take it out. Whereas fatty acid is from acetyl-CoA, but it has to take it out of the mitochondria. Ketone bodies are made when you are under a starve mode. So when you don't have a lot of food and you're going into starvation mode, then the body would say, oh, I better make a lot more ketone bodies. We do make a baseline number of ketone bodies on a regular basis, but it's only activated significantly during starvation. And diabetic too, because diabetic is a form of starvation. You have a lot of sugar in the cell, but the insulin is not secreted, so the cells don't know that they can take the glucose in. So they would stop. And then they would trigger on the formation of ketone bodies, which is acetoacetate, acetone, or beta hydroxybutyrate. These are the three components that qualifies as ketone bodies. All right? And you can actually smell sometimes people that are starving or people that are diabetic has a breath that has high in acetone. And that's because the body is thinking like that. And how did you make the ketone bodies? You start with acetyl-CoA. And it's a very simple process. Why simple? Because you're starving. You can't do very complicated steps. Do you understand? So this is the process. Now remember, did I give you the name of the ketone bodies, the three components? Maybe this is a better slide. If you make acetoacetate, beta hydroxybutyrate, and acetone. Those are collectively referred to as ketone bodies. You gotta know that, okay? They are produced in the mitochondria from acetyl-CoA. And the process of making that is called ketogenesis. And I can give you the pathway. It's so simple. I mean, this is about one of the simpler pathways. Look. In this time, you say, oh, the chemistry is really easy. You just take two acetyl-CoA and suck them together. Usually, I always say, oh, you can't do that. It's too simple. You have to take three and stick two like that. But in this case, you can. You take two acetyl-CoA and you stuck them together to make that acetyl-CoA. And then you combine a third acetyl-CoA and you make this molecule here. 3-hydroxy, 3-methyl-glutyl-CoA. And then you remove one of the acetyl-CoA and you end up with this ketone bodies. This is your ketone bodies. So you actually combine 2, 3, and then you took out one again, and now you end up with this structure here called acetoacetate, sorry. And then you do oxidation reduction step again, and you can convert, the acetoacetate to acetone. Okay. Go another reduction step, and you get beta hydroxybutyrate. So this is ketone bodies. This is another form of ketone body. And then the third form is this form. Alright? So all three are collectively referred to as ketone bodies. And the brain will use this. It will use glucose first. If we don't have glucose, then during starvation it's like, okay, I'll use this. What won't the brain use? What will the brain not use? Fatty acid. But they will use ketone bodies. That's why we have to make it. Now, you know how to make ketone bodies? Then, do you know how to break it down? Same old, same old. This is about the simplest. When you're starving, you really can't expect too much out of it, right? You can reverse the process. You take beta hydroxybutyrate to acetoacetate, and then you break it down all the way to acetyl-CoA. Oh God, I think it's not even long enough. You break it down all the way to acetyl-CoA. This is the simplest two reactions, and this is called ketolysis. So one is the making is called ketogenesis. The breaking down to acetyl-CoA is called ketolysis. So, you know, that's how you would learn all the pathways, right? And I'm done with this. We're done with lipid metabolism. So I got 2 more chapters to cover before the next test, and I recommend that you start studying now. I know we just finished a test, but yeah.